positive feedback, negative feedback, all-to-all feedback, or some spatially profiled feedback. You have it all. Some feedback pathways are plastic, some are not plastic. Here's an example. I showed you this slide yesterday. So you get the receptor input, which just comes here, and we're going to stay at this stage, where things then go off to another part of the brain, and then here's one possible feedback loop. Comes back like this. Okay? And I'm going to focus on this one for the next few minutes. There's a certain delay, because there's wiring to go from one place to the other. So this delay is about 8 or 10 milliseconds. Okay? It's a different part of the brain. So, but notice there's other loops. There's ones that go up here, and then down here, and back here, and back here. There's all kinds of different loops. Okay. So if you put two fish next to each other, you can get beating frequencies, because they're like, tuning forks, or it's like putting two notes next to each other on the piano, right? One guy's at 1,000 hertz, the other guy's at 950 hertz, so they feel beat patterns. Sometimes they're swimming around, and the modulation just becomes some noisy thing. And what we're going to do is we're going to look at how the geometry of the stimulus affects the feedback and the dynamics of the cells, the geometry of the stimulus. Okay, so here's an example where you have a geometry that's very narrow. In the frequency domain, spatial frequencies are high. Fourier spatial frequencies are high. Okay, so it's a localized input. So this is typical for if you're looking at something small with high resolution, like a bug or something. Here's another kind of stimulus, which is when they're next to each other, then they feel a whole body. And the whole body that has much lower spatial frequencies. All right? So we did an experiment. Experiments were done over in our collaborator's lab, Joe Bastian, in Oklahoma. We did the following. If you apply the local stimulus, and you record from the cells, this, by the way, is not the experiment that was done. This is what you would like to do, to record from 100 cells, but you have only one cell. But these days, it's actually been done with 2. But you put a dot every time there's a spike, all right? So here's the activity of the cells as a function of time. If I put the stimulus in or not, if I put this bug in there or not, you wouldn't be able to tell the difference to the eye from that particular pyramidal cell at the next station there. You don't see the difference, okay? Somewhere this bug is encoded in maybe an extra spike or 2 per second in there, okay? But it's there, and as a population it can figure it out and it can go and eat the bug, okay? But I draw your attention now to what happens when the stimulus is full body, okay? In the time domain, this is basically low-pass noise, okay? But in the spatial domain, it changes. And now what you see are frequencies. You see waves going along. So this means that the cell is firing, then not firing, firing, not firing, and so on, okay? So there's some feedback in the system that we know about because Len Mailer figured out this circuitry many, many years ago. And he knows that there's a feedback pathway here, and we know that it's all to all. All the cells here project back to themselves via this bunch of cells, and we know that the feedback is negative. So that means that if we look at the autocorrelation function of the spikes, there should be some inhibition at around 10 milliseconds. That if a spike occurs now, it's not likely to occur 10 milliseconds later because there's inhibition that has to go around the loop. And sure enough, that's what happens. Let me show you what the autocorrelation looks like. That's what the autocorrelation of the spikes looks like. So this is the inhibition at about 10 milliseconds. There's some excitation followed by inhibition. Now, I draw your attention to that first plot where I want to be able to open the feedback loop to figure out what's going on, to see if it's important. So you can do that. Here's the loop. You inject xylocaine, which is what the dentist does to you if you're not brave, and you turn off the feedback pathway, all right? And what happens then? Well, it turns out the oscillation goes away. So here's the curve without the xylocaine, here's the curve with the xylocaine. So the black curve, you see the oscillation is gone. This is the interval histogram. It had a nice second peak because there's an oscillation going on. And now it's back to just one peak. And if you let the drug wash away, then you're back to the original result. So this is telling you two things. It's telling you that the delayed feedback pathway is important in setting up this oscillation. It's also important that it enables the fish to distinguish between two classes of stimuli. Things that are very localized, that don't cause oscillations, and things that are very spread out in space, that do cause oscillations. Okay? I'm not claiming that's the only cue that the fish has to distinguish between bugs and other fish, but it's an important cue because the geometry changes the dynamics. Okay? Does that make sense? So, okay. How do we model this? Yeah?
So how many neurons are involved in this?
It's about 30,000 or so. And the ELL is divided into maps. So, yeah, something like that. The main pyramidal cells, there's a lot of supporting actors, which my experimental colleague keeps telling me, don't worry about that. Don't worry about that. Don't worry about the interneurons and so on. Okay.
So when you change the size of the stimulus, you change the number of neurons which are...
That's right. So that's what, now we want to see if we can get that out of a model. If you have a population of cells and you stimulate only one, you should see nothing. But if you stimulate everybody, you should see something. Now, when you model this kind of thing, you say, okay, so we model the ELL pyramidal cell network as a network of leaky integrating fire neurons, and the membrane potential of the ith neuron looks like this, dVdt's minus V plus the input plus some feedback. Okay, and the feedback, we just put some delay. Now here we have the population. Okay, so if you just code this up on your computer, what are you going to get? Delayed feedback with LIF, spikes propagating back to everybody. You get periodic solutions. So you're going to get fire, so the voltage is going to go up, hit threshold, reset, so on. So you'll just get periodic firing. Okay, is that a very satisfactory model? It's not exactly what we see, right? If you follow one of these cells here, it's not periodic. It's actually random. It's very random, a CV almost of 1, practically no correlations. So this is not a very good